Good afternoon. I wanted to go over with you a few more things from today's lesson so that you could reinforce and study at home. Represented in this picture, I have this area here that houses the DNA known as the nucleus. This is representing your nucleolus. Here I have a double helix of DNA. Here I have a single strand of messenger RNA, and I've highlighted that in orange so that you can keep track of where this ends up going. Outside of the nucleus, you have your rough ER, which is studded with ribosomes. And then there's a section of smooth ER that does not have ribosomes attached. Now in this picture, I do have small little structures here that are representing different amino acids that would be floating around in the cytoplasm of this cell. So let's say that this was a cell, and I have tRNA molecules that have come by and picked up amino acids. So I'm just going to put an AA in this box. Now, the DNA has the blueprint. A single copy of messenger RNA is made, which we call transcription. Now that messenger RNA can exit out of nuclear pores. It's processed first, as in my earlier video showed, and then exits out to the rough ER where ribosomes take over and finish this process. Now, when we get into this discussion on ribosomes, there's a slight confusion of what this is. So we know the nucleus makes rRNA, and that happens here. The nucleolus also makes ribosomes, but they're not finished. So there's protein pieces that end up going into this, and it finishes that ribosome, which will exit back out of a nuclear pore, back down, and that's what you have here in red. So I blew one up over here, and you can see that there's the large subunit, and then there's the small. You can think of this ribosome as a vise. So as the messenger RNA is fed through, and I have that shown here in orange, you're building a polypeptide chain, and you can see that here in yellow. So all these little shapes that I have here were from different amino acids that corresponded to the appropriate codons. So every time I had a codon, there would be a respective anticodon. So to blow this up so that you can see it, if I have a strand of messenger RNA, and let's say that that code was A, U, G, the appropriate tRNA that would match up with this would be U, A, C, and that would be carrying a particular amino acid that would be dropped off and left on this chain of polypeptides. And then this chain would grow until we get to that stop point. Peptide bonds are added in between each of our amino acids, and we have ourselves a primary structure of a protein.